Okay, good afternoon, everybody, and welcome to today's live webinar. Today, we're going to be discussing the outcomes of Run 14, the variant classification EQA scheme. This is an interactive webinar, and I'd also like to thank in advance the organisation and funding from AstraZeneca and Merck for promoting it. Okay, next slide, please. Just a couple of housekeeping requirements. So we've muted your microphones, so avoiding any background noise. We are recording the webinar. We will make it available to you probably about 2 weeks after this date. We'll let you know via the EMQN and GenQA mailing channels, and it will be published on our respective YouTube channels. But most importantly, this is your webinar. It's interactive. We strongly encourage you to support the question and answer facility at the end of it. It will be dedicated to our experts, giving you some feedback on the actual results itself. So please do use the Q&A to actually ask your questions to the experts. Next slide, please. Feedback is also important, so we want to learn from what we've been doing as well as much as you want to learn from what we've been telling you and train in the training side of it. So please do contribute to our feedback survey. You will get an email shortly after the completion of the webinar. It takes about 2 minutes to complete it, and your feedback is important to us, so we will listen to it and learn from it as well. So in advance of that, please do contribute if possible. Thank you very much indeed. Okay, next slide, please. So I'm now going to hand over to my co-chair, Professor Sandi Deans from GenQA, who will do the introductions and the background behind the webinar. Over to you, Sandi.
Lovely. Thank you, Simon. Just like to add my welcome as well to you. So we are joined today by our two experts on our panel who have helped facilitate and support this Run 14 of the variant classification. We have Dr. George Burrell from the Manchester Genomic Laboratory Hub in the UK, and also Dr. Andrew Mariani he's from Edmonton in Alberta in Canada. So it's incredibly early for Andrew to join us. So a special thank you for taking the effort to do that today. So next slide, please. So just to start, to give you a bit of background, these are the disclaimers from the speakers. Next slide, please. A background of the overview. So this is participation in Run 14. So this is about classifying variant in the BRCA1, BRCA2, and other HRR genes. Next slide, please. So the platform that is used to deliver this assessment is the GENIE platform, which is the GenQA Genomic Online Individual Education Platform. So the idea is we have modules here that are specifically focused in different areas of genomic testing and different roles and tasks that are performed within the workforce. You can access it anytime through the GenQA website. It's online and it's available globally all year round. And for some of our scenarios, they're randomly generated, which means you and a colleague can log in and perform these modules, and you'll get different quick cases and questions. So there's no collaboration or collusion. You can also participate multiple times if you wish to. And the idea is you get an automated assessment back to you. And you can do that at your own pace. And there's an opportunity for your managers or your training officers to review performance and discuss that with you. So we use this GENIE platform for the BRCA Run. And now we've gone to Run 14, which is just closed. And I'm now going to go through some of the overarching data. Next slide, please. So we have delivered the run during the calendar month of April. That should be 2025. We had a number of registrants, 446. And as we see with all runs, that we actually have reduced numbers who actually participate. So 359 participated in this run. And they were from 58 different countries. So I think you can see we've got global spread, and the fact that it's online and it's not time sensitive or it has to be worked on during the day means that we get participation globally. The graph along here just demonstrates the number of different countries, and also the one above on the top right-hand side breaks it down by countries where there's more than 10 registrants. So here the top participating countries are the UK, we've got Spain, Poland, and Germany are the most predominantly participated countries. Next slide, please. One of the benefits of collecting the data as well as the classifications from participants is actually we can see which type of guidelines and the application of different guideline combinations are in use. And we can now also map them to previous runs to see if there's any change. I think here you can see that we still have a spread of different approaches being used by different individuals. Here you can still see that the ACMG from 2015 is being applied still by slightly fewer, 27% of participants in the previous run compared to 23% in this run. You can see a slight increase also in the combination of 3 guidelines, that's this ACMG 2015 guidelines, the UK ACGS 2020 guidelines, and the CanVig UK 2020 guidelines. So that has increased from 34% in the previous run to now 40%. So the majority in the run for 14 used this 3-way combination of the guidelines. Next slide, please. We can also see who participates and their expertise, as in self-declared expertise. So for the specialties of participants, you can see that the majority, that's 39%, are actually based within genetic specialty, with clinical genetics following up very closely behind at 30%. And I think this reflects the different approaches and who actually performs variant classification depending on the country or local practice. We know in some countries it sits very firmly within the laboratory scientists' remit, and in other countries it sits very much within the clinical teams. The expertise of participants here as well, you can see that 44% deemed that they were experienced scientists, and the rest are others. And I think maybe we need to look at how we can break this down a bit more granular so we can see what sort of level individuals are operating at. I think what's really nice to see, and I take particular pleasure in this, is seeing that 16% of participants are actually recently qualified scientists, which shows that this is really an excellent training tool and has helped for education and demonstrating competency, not only to the workforce, but those that are newly qualified as well. Next slide, please. So this is a summary of the 6 variants that were included in Run 14, and we will hear shortly from Andrew and George more in depth of the findings and some of the classification issues that have been addressed. First thing to say is we cover both BRCA1, BRCA2, and we've got 2 ATM gene variants in this run. We also want to cover not just a raft of different genes, but also different classifications. So again, you can see here we've covered class 1, class 2, 3, and 5. So the whole raft from benign right through to definitely pathogenic variants. And again, we try and address the issues of using different guidelines as to whether classifications may change. So here you can see for variants 4 and 5, the classification did not change regardless of the guidelines applied, but variant number 3 did. But clinically, due to clinical management, a benign or a likely benign variant is basically handled in the same way. So despite the giving different classifications depending on the guidelines, clinically it would not make much of difference. But we'll hear more about that very soon from Andrew on that variant. So next slide, please. We also like to see what the overall performances of all of our participants. And I think we can very clearly see here that for this run, 14, we had a much, much higher number of participants. 50% of those were correctly classifying all 6 variants. And if you look at the table on the right-hand side, you can see that this by far is the highest percentage of accurate classifications according to our expected classifications. So I think there's multiple reasons for this. Probably the more use and following the guidelines more accurately, hopefully through education and practice, but also potentially with the use of the ATM gene guidelines specifically as well for those variants. So next slide, please. Right, so I'm now going to hand over to Andrew, who's going to kick us off with an in-depth discussion of variant 1. Thank you, Andrew.
Thank you. Next slide, please. So variant 1 is a missense variant in the ATM gene. It changes the serine at position 1589 to a leucine, and it's not in a functional domain. Next slide, please. So this variant is found twice in NOMAD version 4.1. It's classified in ClinVar as being of uncertain significance, and in silico predictions are predicting the variant to be benign, with the exception of CYP. Next slide, please. So using the ATM BSEP guidelines and CanVig guidelines, you can apply BP4 as the in silico prediction. In this case, the Rebel score, which is what the guidelines recommend using, is 0.169, which is below the threshold for BP4, so BP4 is applied. If you use the CanVig specific guidelines, you can also apply PM2 supporting. This variant is present in NOMAD version 4.1, but according to CanVig, it's recommended to use NOMAD version 2.1, and this variant is actually absent in NOMAD 2.1. So you can apply PM2 supporting using the CanVig guidelines. Either way, this adds up to being a classification of a variant of uncertain significance. Next slide, please. So the majority of individuals did get this classification as expected, so classified it as a variant of uncertain significance, with a few individuals classifying it as likely benign. Next slide, please. Of the 312 participants, 312 participants classified this as a VUS, with about 10% classifying this as either a likely benign, or one individual called it pathogenic. Of the 35 participants that classified this as being likely benign or benign, half of them used BP1. BP1 is used for a missense variant in a gene which primarily truncating variants are known to cause disease. And according to the both guidelines, BP1 is not applicable for ATM. So BP1 shouldn't be used in this classification. Next slide, please. Now, with regards to the use of PM2, this gets a little tricky. So our classification was based on the ClinGen version 1.3, which was released in March of 2024. This was the most recent VSEP guidelines for ATM. However, shortly after this, there came a publication from the same individuals that are in the VSEP, and the publication is based on the ATM classification guidelines. And so they actually published an ATM classification guideline publication, which is published at a later date than the latest ClinGen guideline. And when you compare the publication versus the ClinGen version 1.3, there actually are some differences. I'm not going to go through all the differences, but I will highlight the use of PM2. Next slide, please. So in the publication, they specifically recommend using NOMAD version 2.1 as CanVig does for the classification of ATM. So if the variant is absent in NOMAD version 2.1, you can apply PM2 at supporting strength. However, in the ClinGen ATM version 1.3 recommendations, they've removed the use of which NOMAD version to use. We did reach out to this VSEP group, and they did recommend using the latest version of the ClinGen recommendation, so in this case, version 1.3, over the publication. They didn't clarify to us which version of NOMAD was most appropriate, but I have been told that they are discussing which version of NOMAD to use. So in this case, it's a bit gray whether you should be applying PM2 or not. We chose not to apply PM2 because this variant is found in NOMAD version 4.1. However, I do understand why people might have applied PM2 using the VSEP guidelines because it is at the moment a little bit unclear. Next slide, please. And then using the CanVig UK guidelines, it's very clear. It does mimic the VSEP the publication from this group where they recommend using that version 2.1. And all in all, 78% of participants did use PM2 at some strain. In the end, it doesn't change the classification of this variant. And I understand why individuals would use PM2, but at this point, it's a little ambiguous whether we should be using it or not, using the QING and VSEP guidelines. We are hoping to get some clarity in the near future. Next slide, please. And now I'll hand it over to my colleague, George.
Thank you, Andrew. So this second variant is also an ATM variant. It is a deletion between between 6415 and 6416, which results in a frame shift and premature termination of the coding DNA. The variant is also found in Nomad version 4 5 times. It has multiple classifications on ClinVar, mostly pathogenic, and also has 2 entries on LOVD. So this is the criteria that we applied. So using the VSEP criteria, and the CANVIC VSEP and CANVIC criteria, we applied PVS1 very strong, PM3 with a variety of strength, and I will go through that, and PM5 PTC at supporting level. Using the CANVIC criteria, as Andrew just mentioned, there are some differences currently between them. PM2 supporting was awarded for this variant as well. Overall, using both guidance and despite the different strengths that have been applied for PM3, the classification of this variant is pathogenic. Most of the participants, as you can see from this chart, got it as expected at class 5, some at class 4, and very few at class 3 or 2 or unable to classify. So PVS1. So that's relatively straightforward. The ATM VSEP guidance provides a detailed PVS1 flowchart that you can follow. You can find it in their page on the ClinGen website, and our variants fall into the region between serine 2 and glutamine 2979, and so it is predicted to undergo NMD and can get a PVS1 very strong. For PM3, it gets a little bit more complicated. So again, the guidance provide a very nice summary of what can be applied when, whether a phenotype is confident or consistent, and different levels, and they also list the phenotypes that can be used to determine whether a phenotype is confident and consistent. This variant appears in 3 publications. The first 2 are listed here. They do actually refer to the same patient, so we can only use one of them, and we went for the original one published in 1996. And you can see the variant here alongside another variant in patients with AT. This just to highlight that this is an old nomenclature presented here. Now, if you follow the criteria, the publication states that these mutations listed in this paper has been identified in families with AT. And based on the description from the VSEP guidance, it states that if a publication claims a clinical diagnosis of AT, then that's taken as a face value of confident diagnosis. They do, however, provide the phenotype criteria that you can use yourself to assign whether a phenotype is confident or consistent, and that criteria can be used if a publication also list the criteria or the symptoms of the patients. So in this case, the paper did list the symptoms of the patient, which was mainly very mild. And so if you decide to not to follow the first statement that if a paper tells you there's a clinical diagnosis, use it as a face value of confident diagnosis, and actually look at the specific phenotype they provided and compare that to the clinician list of phenotype, you might end up with a consistent phenotype rather than a confident phenotype. Or if you take it as a face value of confident diagnosis, you will have a confident diagnosis, and the difference will result in either a supporting PM3 or a moderate PM3. I will mention in a minute what we decided on. But then the third paper, which is listed here, is another paper with this variant, and they have done a functional analysis and showed that in this case, there were no ATM protein detectable. This paper does reside behind a paywall. It's a subscription only. And we realized that many people don't have access to the subscriptions, and so we did not include that in the assessment. So if you use a phenotype confident from the first two papers, with an unknown phase, then you end up with 2 points and PM3 moderate. If you do use the detailed phenotype and the symptoms that provided in the paper and decide to use it as the phenotype consistent with a phase unknown, you end up with a 1 point, and that will only give you a supporting level of PM3. We have gone with the VSEP recommendations of using it at face value and considered this as a PM3 moderate mainly. So these were the classifications that were submitted. Again, excellent performance from the participants. Nearly 90% of the participants got it as pathogenic, and nearly 98% got it between pathogenic and likely pathogenic, which is a safe classification. You can see that there has been a variety of PM3 strengths applied. So you have some applying at supporting level, some at moderate level, or most at moderate level, and some at a strong level if they had access to the second paper. There were few classifications of VUS. Some haven't used PM3, some have misused PVS1 at a strong level, and a couple did not apply PM5 PTC. And just this is just in case we had this also as a PM2 clarification, but Andrew covered this nicely. Essentially, we still have a little bit of mismatch between the CANVIC guidance and the VSEP guidance, but in CANVIC, as soon as the VSEP update is published on the use of NOMAD version 4, the latest version, we will be updating the CANVIC guidance to match that. I'll hand back over to Andrew. Thank you.
Thank you. Next slide, please. So the third variant is a missense change in BRCA1. The change is an arginine at amino acid 170 to a glycine. It's a missense, and it's not in a functional domain. Next slide, please. So this variant is present in NOMAD version 4.1. It is not reviewed in BRC Exchange, and it has conflicting classifications in ClinVar. In silico predictions, predict this to be a tolerated change. Next slide, please. So this is an interesting variant because depending on which guidelines you follow, you weigh the evidence differently. So according to the VSEP and CanVig guidelines, you can use, according to the VCEP guidelines, you can use BS1 at supporting, EP1 at strong, and BS3 at strong. And that gets you to a classification of benign. Whereas if you use the CanVig guidelines, you use BP1 at supporting and BS3 at moderate, which gets you to likely benign. They are slightly different classifications, but they are in the same, they have the same general clinical outcome. And I'll get into the differences of these two guidelines in the following slides. Next slide, please. So most individuals did classify this variant as expected, either class 1 or class 2. However, there were a significant number of individuals who also classified this as of uncertain significance. Next slide, please. So for the use of BS1, using the ClinGen version 1.2 recommendations, it's recommended that you use NOMAD version 2.1 non-cancer exome only, and version 3.1 non-cancer exome only. So based on this, in NOMAD version 2.1 non-cancer exomes, the group max FAF is 0.000384, and this is coming from the non-Finnish European, so it's not a founder group. And based on that, you can apply BS1 at a supporting strength because the group max FAF is above 0.00002, but less than 0.0001. So they have a tier, they've broken down BS1 into strong and supporting based on the frequency in the population. And this variant would fall under the BS1 supporting. The CanVig guidelines do not break down BS1 in this manner, and so therefore this variant, you shouldn't use BS1 using the CanVig guidelines. Next slide, please. Oh yeah, and this was, so CanVig has a slightly different classification for how you determine whether BS1 is appropriate. And based on the frequency in the population, the maximum tolerated allele count would be 96. And in this case, in females, we only have 7, so it doesn't meet the threshold for using BS1. Next slide, please. So now for the use of BP1. BP1 is a missense variant in a gene which is primarily truncating variants are known to cause disease. In both the ClinGen and CanVig UK guidelines, BP1 can be applied to variants in BRCA 1 and 2 that are not found in key functional domains. Using the ClinGen guidelines, BP1 is applied at a strong strength, whereas using the CanVig UK guidelines, BP1 is applied as supporting strength. They are looking at the same key domains. This variant is not found in a key domain, and therefore BP1 is applicable, either at strong or supporting, depending on which guideline you are using. Next slide, please. And the third piece of evidence is the functional studies. So this variant is published in a functional study using the ClinVar, the VCEP guidelines. You can actually go to table 9. It's one of the supplemental tables, and you can search for this variant, and the VCEP tells you exactly what strength to use it at. So in this case, it's BS3. CanVig is using the same publication, and if it were a evidence towards being pathogenic, you would also apply this at a strong strength, but they've weighed this publication or evidence towards the variant being benign as only moderate and not strong. So this is another difference between the two guidelines. Using the ClinGen guidelines, this is a strong piece of evidence towards it being benign, whereas using the CanVig UK guidelines, this is a moderate piece of evidence towards being benign. And this is all based on the same publication by Storita et al. In 2018. Next slide, please. And so overall, 112 participants, so about a third of participants did use PM2 at some strength, even though PM2 wasn't appropriate. In some cases, BS1 was appropriate, depending on which guideline you're using. As well, about 25% of participants used BP4, while 36 of those also used BP1. So BP1 and BP4 shouldn't be used together. BP4 is if you're using in silico predictions to as evidence for a variant being benign. However, since this variant wasn't found in a key functional domain, you shouldn't use BP4. You should be using BP1 instead. Using BP4 and BP1 together is sort of double counting evidence. It's also not recommended in the guidelines. Next slide, please. And now I'll hand it back to my colleague, George.
Thank you very much, Andrew. Excellent. Sorry, the slides went slow. So the next variant is a missense BRCA1 variant affecting leucine 512, and it's not present in a functional BRCA1 domain. So the variant was present 190 times on NOMAD version 4. It has been reported on BRCA Exchange and ClinVar with a variety of classifications of benign, likely benign, and VUS. And there were various in silico predictions. So this variant is classified as a benign variant, and these are the criteria using the VCEP guidelines, which we'll go through in details. And these are the slightly different criteria that can be applied using the CANVIG guidelines, but reaching a same classification of a benign variant. So to start with, BS1 is applied using CANVIG following these rules. You have to look at the maximum tolerated allele frequency, which has been computed for BRCA as 0.0001 for BS1 and 0.1% for BA1. And CANVIG dictates the use of female controls when determining the maximum allele counts or filtering allele frequency for any variant. So in order to calculate the maximum allele count, we use the CardioDB calculator. The link is there on the screen, and you can see that using the maximum maximum tolerated allele frequency and the number of alleles at this variant, you get a maximum tolerated allele count of 96, while there are actually 111 female cases listed on NOMAD, which is greater than 96, and that's why this variant you can use BS1 strong following the CANVIG criteria. Following the VCEP criteria, which recommends the use of filtering allele frequency, this variant falls in the range between 0.002% and 0.01%, as you can see from the screen, and so it only gets BS1 supporting. BS3, which is the functional analysis. So this variant was listed in Bowman et al., this brilliant paper that did functional characterization of multiple BRCA1 variants of uncertain significance. This is just a summary of the functional studies that's was done in this study, but you can see that they actually use 3 different assays to reach a functional characterization. They use a cisplatin PARP inhibitor and DRGFP HRR assay. And this study has been assessed using the SVI scheme for BS3 criteria strength. This is the Burnett et al. Paper from 2020 that provide this nice and detailed workflow of how you can assess a functional study. So if you follow this workflow, you can reach a BS3 or PS3 strong using the Bowman criteria. If you're not familiar with this yourself or don't want to do assessment, or you don't need to do the assessment for all the studies, we previously covered that in our in some of the talks for this scheme, but essentially CAMVAR UK, which is the variant database in the UK that also published the CAMVIG guidance, you can see some of the information or for the variants. So the variants, if you head to the CAMVIG website, they are annotated with various things, including functional studies. So if you go to this variant and go to the functional analysis tab, you will see the results of the Bowman et al. And you can link to a CAMVIG functional study scoring sheet, and you can see that the study has already been assessed and it reaches a strong level of evidence. That is applicable for both BSEP and CAMVIG as BS3 strong. Moving next to BP1, this achieves strong using VSEP and only supporting using CAMVIG. We have come across this before between CAMVIG and VSEP, where BP1 and BS1 seems to be on the other side of the strength. They balance each other out, but it's a difference between the guidance to be aware of. But this variant is essentially located outside the functional domains in BRCA1 that are listed here, the ring domain, the cold domain, and the BRCT repeats. And because of that, using VSEP criteria, you can apply that as strong, while in CAMVIG, you can apply that only at supporting. Please note that for any variant to achieve BP1, when it's outside this functional domain, it has to have no prediction to impact splicing. And in this case, this variant has no predictions to impact splicing using Splice AI. Now, for the other criteria that we can apply for this variant is BP5. So BP5 in the original guidance, as many of you know, was using expert groups, but this has been labeled or relabeled, and you can use it now using any published likelihood ratio studies. And there are multiple studies published for BRCA1 and BRCA2 that show segregation, co-occurrence, family history, case control, and combined likelihood ratios. Again, using CAMVAR, you can see a summary of these studies in BRCA1 and BRCA2. And for this variant, the Parsons combined likelihood ratio allow you to use BP5 at a strong level, following both the CAMVIG guidance that you can see in the table at the bottom, and also following the VSEP guidance. So that's BP5 strong. Oh, I lost the clicker. So if we can move to the next slide, thank you. So you can see that most participants achieved a benign class 1 as expected, few at class 2, and also few at class 3. Next slide, please. So this is a summary of the classifications that were submitted. And you can see that 254 classified as benign, so that's 72%, and more than 88% achieved a very safe classification of benign, likely benign, which is something that will result in the same clinical implications. So that's why we think of them as safe. The main reason for achieving likely benign was some people using PP3, which I will cover in a minute, and the same reason for the main reason for VUS was the application of PM2 and PP3. There was one report of likely pathogenic, but that was through the incorrect application of multiple criteria. I have the clicker back, so I can control it now. So just to clarify with PP3, because a significant number of participants applied PP3 when it shouldn't be applied. There is a list on the VCEP side and also on the CAMBIC criteria of the BRCA1 functional domains, and they're listed here on the screen. And you can only apply a PP3 if the variant has in silico predictions and is present within these domains. Now, this variant in this case is outside the functional domains. It does achieve a high level score of 0.716, which would have been appropriate for PP3 if it was within the functional domain. It doesn't even achieve 0.28 for base del, which is the criteria used for in silico prediction by the VCEP Enigma guidance. So it shouldn't be used, but it is relatively high in silico prediction, and that's why probably more 113 participants applied it. But please remember, it's only applicable if it is within the functional domains. We'll move over to the next variant, which is also I'm covering. So this is a BRCA1 duplication that results in a frame shift and premature termination. This variant is absent from NOMAD. It is present on BRCA exchange, and there are multiple submissions on ClinVar, most of which are pathogenic. So this variant achieved a class 5 pathogenic variant class using the VCEP, and these are the criteria that are applicable: PVS1 very strong, PM2 supporting, and PM5 PTC at strong level. Again, a difference with the CAMBIC criteria, the same 3 from the VCEP, but on top of that, you are able to apply PS4, and we will go through that now in details. So again, it's excellent to see that most participants achieved the expected class 5, some class 4, and very few class 3 or unable to classify. So PVS1, again, relatively straightforward. If you open the VCEP guidance and you head to the appendix, you will be able to get the workflow that explains, or the flow chart that explains exactly how to apply PVS1 for BRCA1 and 2. And in this case, this variant achieved a PVS1 very strong. It will result in nonsense mediated decay. PM2, the variant is absent with a good coverage, so it achieves a PM2 supporting using VCEP, but using CAMBIC that can be up to moderate level. So that's a discrepancy between the two guidance. PM5 PTC, again, this is a criteria that can be used for protein termination codon in certain exons. We've used it earlier in ATM and in using the VCEP guidance and CAMBIC matches in this area. If you go to the specifications tables, you will get also a detailed information on how to apply PM5 PTC, and in this case, it achieves PM5 PTC strong. Now, PS4, that's the difference between both guidance in this setting. So again, in our last presentation, we presented CAMVAR as a resource for variant interpretation, and we have explained that if you register, and people who work in diagnostic labs can register from all over the world. It's heavily used by UK scientists. And if you log in after registration, you'll be able to access a little bit more information than what's available, including case counts from a UK-based data set. And if you do that and get these cases, you will see that this variant has been seen few times in the UK population. And so following the CAMBIG criteria, the NHSD data, which is the data that I've just shown, does allow you to do a PS4 case control analysis. The controls should represent appropriate ethnicity and sex. So we use female cases from UK Biobank. And it's because most of the cases that's for controls, because most of the cases that are present in the NHSD data are female affected cases. We apply a Fisher exact test, and based on the odds ratio that you can get, you can apply PS4 at various strength. In this case, in order to get our control population, so we've seen the case population earlier. To apply now the control population, you need the count from UK Biobank. To achieve that, using the data that's available on Nomad, you can get Nomad version 4, look for a nearby variant location, look for Nomad version 4 non-UKB, and then if you subtract them from each other, you will get the number of cases or the number of controls from the UK Biobank. And then if you use an appropriate case control analysis, I'm really sorry, the clicker is lost again. If we can move to the next slide, thank you. You can actually apply PS4 very strong for this variant. So in this case, we have used the PS4 calculator that's published by the CAMBIG group. The link for it is at the bottom of the screen if you haven't seen it. And you can see that this variant achieves a very strong level of PS4, which allows us to use PS4 very strong. That option is sadly currently not available if you are using the VSEP criteria. Next slide, please. So overall, most participants, 95.7%, applied or classified this as pathogenic. More than 99% classified as pathogenic or likely pathogenic. There have been VUS classifications, but in this case, they do not apply PVS1. Those who have, or some of the participants who classified as likely pathogenic, did not apply PM5 PTC. This is a criteria we know from experience that a lot of participants are less experienced with. So I do recommend looking for the details on the VSEP resources to familiarize yourself with PM5 PTC. 128 labs used PS4, only 22 as very strong, but most of the labs or participants who applied PS4 were following the CAMBIG guidance. Next slide, please. And I can now hand over back to Andrew. Thank you.
Thank you. Next slide, please. All right, so the last variant is a frame shift in BRCA2. It's a deletion at nucleotide position 9097. Next slide, please. So this variant is absent in Nomad version 4.1. It's classified as pathogenic in the BRCA exchange, as well as in ClinVar and LOVD. Next slide, please. So using either the VSEP or CANVIG, you can apply PVS1 very strong, PM2 at either supporting or moderate, and PM5 at strong, which gives you a classification of pathogenic. Next slide, please. So the majority of individuals did classify this as expected, which is great to see. Even those who didn't classify it as pathogenic at least got it to likely pathogenic, so it is in the safe zone in that a clinical outcome would not change based on the classification. And so this is really good to see. Next slide, please. I'm just going to go through the evidence we used here. The first piece of evidence is PVS1, very strong. So following the PVS1 decision tree, this is fairly straightforward. It's a frameshift variant that is upstream of the 3309 amino acid, so it is applied at PVS1 very strong. Next slide, please. The next two pieces of evidence, I'd say about half of the individuals did not apply the evidence or didn't apply it appropriately. So in this case, I think it's equally as important the conclusion you get, but also how you arrive at that conclusion. So I'm hoping everyone got to pathogenic or likely pathogenic by using the correct evidence. If you got to it by another mechanism, then it might not necessarily translate to another variant. So even though everyone got to pathogenic or likely pathogenic, it's not always clear the route they used to get to this classification. So for example, 45% of individuals did not use PM5. I know PM5 has been repurposed. It wasn't initially intended in the 2015 guidelines to be used for truncating variants, but both for ATM and BRCA, this code has been repurposed to apply to protein truncating variants. And in this case, it's applicable at a level of strength that's strong. Next slide, please. The second piece of evidence people did not, about half of the individuals did not use is PM2. And we're trying to figure out why this is. Often what we've seen is PM2 is perhaps overuse, so it's a little less common to see PM2 not being used for a variant when it's applicable. And so there are a couple of explanations we have come up with, and we're not entirely sure which one is correct, and it might vary on the individual. One possibility is that there are a couple of other frameshift variants at this position, and I believe at least one of them is present in the NOMAD database, so it's possible that this frameshift was confused with another frameshift at the same amino acid position. The other possibility we came across was this variant is seen 3 times in the NOMAD 2.1 database. However, this is only if you're looking at including samples that did not pass the quality filters, and I'll get into what I mean in the next slide. Next slide, please. So if you search NOMAD for this variant, it's absent in version 4.1, and it's generally absent in version 2.1. The default setting is to not include samples that failed quality control filters. Next slide, please. However, if you do include samples that did fail quality control filters, you do see this variant 3 times in the NOMAD version 2.1. And you can tell it failed filters because you do get a warning. Under the exomes, you see this warning flagging this as having failed the quality control filters. One observation I made was there are some commercially available software that when they search or link out to NOMAD, they will link out to the NOMAD with the poor quality filters included. So if you link out using a commercial software, you might actually see this variant as being present in NOMAD version 2.1. Whereas if you apply the appropriate quality filters, which is the default setting by searching in NOMAD, this variant is absent. So it's also possible that individuals were looking at these 3 allele counts in NOMAD version 2.1. However, these 3 alleles did fail quality settings, and if you remove the poor quality reads samples, then these 3 alleles disappear, meaning that this variant is absent in NOMAD. Next slide, please. And so, yeah, that's all I have for this one. Thank you.
Great. Thank you, Andrew, very much indeed. So, and to George for going through it in great depth. Actually, it's great to see the standard improving quite considerably from previous runs. And I think that's demonstrated here by our bubble plots, which comes together the data from all 6 variants. So the larger the bubble is indicates the proportion of individuals that provided that classification. So the larger the bubble is, the more individuals said that that was the class of that variant. The green bubbles are the expected classifications, and red are those that are not expected. So I think very clearly and visually you can see here that the red bubbles are much, much smaller than the green, indicating that more individuals are coming up with the correct classification or the expected classification. Next slide, please. So for those who did participate in run 14, the summary report is currently being prepared, and we'll have a lot of this information there and much, much more for you. And that will be available towards the end of July, beginning of August. We are currently in preparation to deliver run 15 during early autumn as per our normal calendar, and all the key dates and information will be advertised as usual on the GenQA and EMQN websites. So please do look out for that information. Also, just to say that we've got fantastic questions and queries coming in that we're going to address live now. If we have time to cover them all, hopefully we can, but we'll also give you some written feedback to those that have currently already been answered in the background as the webinar has gone on. So that information will be made available as part of the summary report going forward. So now, Simon, can I join you to ask our lovely panel and Andrew and George to clarify some of the questions that are coming in that are really covering all 6 variants. It's great to see. So again, if anyone wants to submit a question, please pop it in the Q&A function box, and hopefully we'll have time to go through all of them. So Simon, do you want to kick us off with some of the questions?
Yeah, I will do, Sandy. Thank you very much indeed. So George and Andrew, prepare yourselves. Here we go. So we have a couple of questions. I'm just trying to group them together by the different variants. So variant 3. So I think this was Andrew, if I remember rightly. So first question is, can CANVIG guidance for variant 3 meet the criteria for PM2 supporting, as it's concerning that using alternative guidance, it only meets BS1? Was this discussed or considered in the review?
The criteria for PM2 supporting. I don't recall us discussing this. George, are you familiar with?
I'm just checking how many times it has been seen in.
So I had looked through the slides. I couldn't see any mention of that.
No,
so
I don't.
The discussion.
I'm just quickly checking why it was deemed applicable for supporting.
If it requires us going through the slides, what we'll do is we can answer that directly afterwards.
And we'll send the criteria. Yeah. I don't remember us having to use PM2.
Okie dokie. All right. Okay. So I've got another question for variant 3. Can we also consider PP4 moderate, ACMG logs likelihood ratio 2.9? So they're referencing Parsons 2020. So I think that's a question: why can we use PP4 moderate as supporting evidence?
That is something we discussed. Once again, can we refer back?
We'll pick it up.
The report. I don't recall why we didn't use that, but it is something we discussed.
Yeah. Okay. We'll pick that up. There is also a second question. Somebody else has asked exactly the same question, so we're going to answer that in the main report. Sandy, can I hand over to you while I triage a few more questions for?
Yeah, that sounds great. We had one for variant 4. I'm just trying to find it right now. Or variant 6. Let me see. There's one here for variant 6. So Andrew, I think this is your one.
Sure.
So can PS4 strong be applied? And there's a reference here given which states 5 affected individuals with the variant of interest and 665 affected individuals in NOMAD version 2.11 East Asian.
I would have to.
Yeah, we'll have to look into the paper.
Yeah, yeah. I mean, this is great that we're getting such feedback.
Yeah.
It shows the vast amount of resources that are out there, and you can't look at absolutely everything in a, you know, if you were doing it as a research project per patient, you could, but this is great, and we'll definitely have a look at that and then come back.
I'll throw in a more general question while we're triaging a few more, but I think it really relates to just the sheer breadth of guidance. So we've got a question here: will we be harmonising CANVA UK guidelines with Enigma?
I can comment on that, Simon. When CANVIC published the original BRCA1, BRCA2, that was before the VSEP were released. As soon as the VSEP guidance were released, the steering committee, the advisory group for CANVIC, we reviewed the VSEP guidance, and we, in most variant criteria, there is a really good harmonization. The two main areas that we need to harmonize are the level of strength for certain elements. So like BS1 example that we have seen, both are based on really good, strong evidence. So that's a difference to be harmonized. But the other important one is PS4, and that's why we chose a criteria to present here that will, or a variant to present here that will impact that criteria. So the case control analysis, for the ClinGen group, they did not consider the UK case data for them, a data that can be used as a case control analysis. We have presented that to them previously, but in the UK, because we have the NHS data available to the labs or registered users for the CAMVAR, and we do have what we feel is an appropriate control set, which is from the UK Biobank population. We still felt the need that this is such a good resource of data that we should be using, and so we maintain the use of PS4.
Thank you, George. I think that's a great explanation. Sandi, over to you for the next question.
Yeah, lovely. George, I'm going to stick with you. There's one about variant 5. So PM2 through the VSEP specification says, do not apply for insertion, deletion, or indel variants. Could you please clarify this? And then we've also got another comment that's very similar. So EQA guideline states don't use PM2 for deletion or small insertion variants. Why can we use it for variants 5 and 6?
I'm just checking now. Sorry, I need to remember. So variant 5.
Variant 5, yeah. This is awful. We have to keep putting you on the spot.
Yeah, no, don't worry.
Yeah. Again, and we can always update the.
Yeah, we will follow
afterwards.
that, because we can access certain data, but the PM2 was used using the CANVIC criteria, not the VSEP criteria. So again, highlighting another area of difference. So
again, it depends which combination of guidelines or standalone.
Yeah, I'm looking at PM2 moderate using CANVIC.
Yeah,
it's listed also at supporting using VSEP. So we will review that, and we will come back with comments at the scheme at the end.
I do see the VSEP. Sorry, the VSEP comment does say, I'm seeing it, not to use it for small insertions, deletion. So we may have used it erroneously. We'll correct that if that's the case.
Great. There's also one here, Simon, about the BRCA1 variant. About and quoting another paper, so I think we'll put that into the bracket, that we'll take that away, look at that quoted paper and come back.
Yeah, I think that paper's the Parsons et al. Paper, Sandi. So
is it?
I think it's the same. So yeah,
same one. Lovely. So we've covered that. Excellent. Okay, Simon, anything else you want to pick up right? This
I've got, again, just to give George and Andrew a bit of a break from the more detailed questions, just a general comment, really, because it's clear, I mean, from the presentation, and you presented particularly variant 2, you presented an example where you've got publicly available publications and one publication that's hidden behind a paywall. And certainly from my perspective and looking at the data, there's a clear shift in strategies from authors to try and publish in open access. How frequently do you come across problems associated with papers hidden behind paywalls, and how does that affect, you know, the implications for downstream classification of variants? Do you have any? Is that a frequent problem for you, or is it you know, generally isolated?
So I don't, but from my experience personally, I don't see it as much in the cancer realm, but in our cardiac genetic testing, we see a lot of publications behind paywalls.
Yeah.
And we do have access to certain journals that are behind paywalls, and even with that, we're still inhibited to some degree. So yeah, it is actually a fairly large problem depending on which. That's my experience, at least.
Yeah. And I think maybe that's where specialist interpreting labs come into the room, when they maybe have a research interest in that area as well, that they will automatically have access to those pertinent publications. So maybe there's something there about recommending, you know, in a true clinical case, to reach out to colleagues in that area of expertise to get that just little bit more information there.
Yeah.
Okay, thank you guys for that comment. So I've got, again, a more general comment, but related obviously directly to the variant classification. So where we can get LOVD multifactorial data, should we be using this? I can't see that it's being used for any of the cases for the Enigma guidelines.
So I need to look at the LOVD data, but in general, we use the certain papers that have been assessed and reviewed in terms of likelihood ratios. So we're very selective in using them. Andrew, what are your thoughts?
I would agree. I haven't used the LOVD data myself in that manner.
Okay, so it's something maybe we could look back and have a discussion about and feedback.
Definitely. I think that's maybe something we can cover in the report. Yeah. Okay.
Simon, so we've got a very pertinent question here that we keep asking every year. When will we be able to use the new version of this ACMG
You're not allowed, you
guideline?
weren't supposed to include that question. You knew that was the case.
I know, that's going to be released in the next year, because it will change the whole system, and how can we adapt? The point I was going to make is this is exactly why we run these types of assessments and modules, and that it's not about performance monitoring, it's about education. So what we're hoping to do is to run a series of webinars and maybe work through examples with our expert panel once the new guidelines are issued, so we can help and support you implement that going forward. So I think that's sort of the message here. I don't know when exactly they're due, but I do know they're all out being tested at the moment. Is that the case? Andrew, George, any comments?
Heard they're being piloted.
They are in testing. I've seen version of them. The problem is that there will be a lag between the criteria released, then the criteria being reviewed by the various groups. As you can see, the different criteria are released many years after the original guidelines. I sit on a couple of VSEPs where we are still forming criteria at the moment, knowing that we will have to start reviewing the criteria and adapting it once the new guidance is released. So there will eventually be a gap where the version 4 is released, but the VSEP criteria haven't moved over to that. So we will maintain using the most available gene-specific criteria until the criteria are adapted to version 4. So it will keep us all very busy.
It will, definitely.
Absolutely.
Andrew, any comments from you?
No, I agree with what George has said. There will be a lag between when it comes out and when the VSEPs review everything. So I think it'll be a little while before we're applying them ourselves.
And we're going to have that sort of blurry phase as well, when all the databases and all the variant classifications has been performed on old guidelines. And it'll be interesting to see how many actually change classification, and then what impact that will have on clinical services going forward. Yeah, it's a bit scary.
Yeah. Looking at the questions, we've got a lot of questions that are really, really detailed, and I don't think it would be appropriate to go into all the and putting George and Andrew on the spot specifically with the really detailed questions. Sandy, is it worth just reminding the participants on the call, just of the dates associated with the publications and the next steps before we go into the details and close it, wrapping up?
Absolutely. So the Run 14 is currently in preparation, the EQA summary report, with all that information of which pieces of evidence and criteria have been applied at which strengths, and all that information will be put together and reviewed, and hope to get out to you by towards the end of July, hopefully, or beginning of August. And then we are currently preparing for Run 15, which will be open in the autumn. It's normally around September, October time. So we will get some specific dates out to everyone as and when they are finalised. And we'll also notify you through our newsletters and normal communications channels through GenQA and EMQN. But if anyone has any further specific questions, then please do let both EQA providers know, and we'll answer all of the questions that you have submitted as part of that EQA summary report. Obviously, some of them are, as Simon said, are very specific, and we'll go back and look at the manuscripts that you have referenced there, so we can give you tailored feedback to each query. So thank you for all the fantastic questions coming in.
Thanks, Andy. George and Andrew, do you have any question or anything you want to say before we wrap up the webinar?
No, we can address. I was going to address a couple more questions, but I think better to address these detailed questions because they are very nicely detailed, which shows a great level of understanding.
Absolutely.
And as Andrew highlighted, from maybe the insertion deletion with the VSIP criteria, we are listed as an experts here, but we also learn a lot using doing these assessments, and so the feedback and the questions are very, very excellent, and we will answer them all hopefully in details in due time.
And I think following on from that is we're always looking for experts to join our panel. We do things by consensus and taking a lot of points of view. Not everyone always agrees. So we have really fruitful discussions coming up to the classification. So if anyone feels that they are competent and confident to be able to help support the delivery of these modules, then please do get in touch with us. We'd love to hear from you. Yeah.
Totally agree, Sandy. We're all scientists after all. At the end of the day, debate is part of our scientific training.
Absolutely.
Absolutely. Right. I'm gonna wrap up now. So just to thank all the participants, all you guys for dialing in. Also, thank you for those of you who contributed and actually participated in the EQA as well. I want to do a special thanks to George and Andrew for all their hard work associated with not only delivering this webinar, but delivering the EQA in terms of supporting all the assessment, etc. Great job. Thank you for that. A big thank you to the organising team as well. And finally, a big thank you to my co-chair, Sandi Deans. And we'll look forward to catching up with you sometime in the near future with the new webinars in the autumn. And lastly, please do contribute to that feedback participation survey. It makes a big difference to us. There's the link there for you, and you will be receiving an email shortly afterwards. So thank you very much all. Have a good afternoon, and we'll catch up with you sometime in the autumn.
Thank you everyone, and thank you to Angie as well for all her support delivering this too. Thank you. Bye-bye.
Bye for now.